Hello everyone. I will talk about MLR. MLR is a method of making mixture culture of lymphocytes. These lymphocytes are genetically different. By using the functions of MLR, we can predict how the graft rejections of acquired immunity occur. So, I will explain the function and practical use of MLR. First of all, I will talk about the mechanisms of graft rejections. MLR will occur if alloantigens are recognized. Most important alloantigen is allogeneic MHC molecules of a graft can be presented for recognition by the recipient's T cell in two different ways. That is direct or indirect. Direct recognition of MHC alloantigen occurs when alloreactive T cells directly recognize donor MHC and bound peptides on dendritic cell or other antigen-presenting cells. Recipient's CD4 positive T cells or CD8 positive T cell directly recognize donor MHC class 2 or class 1 each, and then they are differentiated into helper T cell or cytotoxic T lymphocyte. Cytotoxic T lymphocytes directly recognize same donor MHC peptide complexes on graft tissue cells and kill them. In indirect recognition of MHC alloantigens, recipient's antigen-presenting cells take up and processes donor MHC molecules. Then, peptide fragment, including allogeneic MHC's polymorphic amino acid, are bound to recipient's MHC and presented. In this case, induced donor MHC specific helper T cells have B cells to produce donor MHC specific antibodies which injure graft cells. Helper T cells recognize same donor MHC peptides bound to recipient's macrophages in graft. Then, they are activated in graft and injure with inflammation. How does MLR occur? Lymphocytes have MHC on their surface, and genetically different T cells recognize the MHC and bring about the immune reaction. Next, I will talk about practical use of MLR. MLC, mixed lymphocytes culture test, has been used to find the mechanisms of graft rejection. Immunosuppressor can suppress the immune reaction in MLC. So it is used as a screenings of immunosuppressor. MLC is used to reproduce and expect immune response against the graft in vitro. Therefore, MLR is very important because we can predict the graft rejection in organ transplant through it. The utility of MLR gives rise to its historic values in immunology as well.